So once a primary tumor is surgically removed, often there's sites of metastasis throughout the body. You don't know exactly where those other tumors exist or the properties of those tumors. Very difficult to know which drug the patient should be administered to kill that residual cancer. But what we do know is that those sites, that residual tumor at the sites of metastasis, they release cells that go into the bloodstream and circulate in the bloodstream. And they exist often in very limited quantities. And so the challenge is, how do we develop devices that can capture those cells and then release them so that we can study the tumor cells and determine which drug can kill those sites of metastasis? One of the great challenges is to develop devices that you can flow blood through at high volumes to capture these cells, which typically are quite rare. And often the cell needs to be captured at the surface. So a cell that's moving, even a human hair away, will never be captured. So we turn to nature for inspiration, and we asked what creature in nature can capture things at a distance. And we turned to jellyfish. And jellyfish have these long tentacles that extend far from the main body to capture food and prey. We developed a surface of a device that had long tentacles, just like the jellyfish, but we made it out of DNA. And we selected DNA that could bind to the surface of cancer cells. So we had these repeat units over and over and over again that could extend very far away from the surface of our device. And we were able to show with that system that we could capture cells in blood at 10 times the flow rate of other devices that had been proposed. And we could do that with the same high capture efficiency. With that project, right now we are moving towards patient samples and we're looking to advance this to the clinic as quickly as we can. We're interested in cell therapy, and not just cell therapy, but more specifically stem cell therapy. I think there's great opportunities because we've reached a time where we can obtain almost any cell type in unlimited quantities. The challenge, though, is how do we get those cells to the right location in the body and make sure that they perform the function that we have envisioned? To address this, we've been developing a number of bioengineering technologies where we control the surface of a cell so that after you infuse that cell, let's just say simple intravenous infusion, we've been able to decorate the surface to target a distant site in the body. So envision that somebody has peripheral vascular disease in one region of their body, and we want to target stem cells there to promote new blood vessel formation. We know the zip code of blood vessels at a site of inflammation, and just like GPS of your car, we essentially can program an address onto the surface of a cell, administer it systemically, and have it target that distant site. We've also gone beyond just targeting cells in the bloodstream to trying to control the cells, how they respond to their local environment, what they secrete, what cell type they become if they're in an environment for a certain period of time. To tackle that problem, we have figured out a way to load cells with devices, very small devices that are particles that contain small molecules. And these particles can be internalized into the cells, let's say outside of the body. And as the particle degrades over time, it releases the small molecule inside the cell for weeks. So this is an opportunity to control cells from the inside out. So you can systemically administer cells, and regardless of where they go in the body, we can exhibit control over them by delivering these small molecules inside the cells to target specific pathways to get cells to boost secretion of an important therapeutic factor or to control what cell type that stem cell may become inside the body.